The overall goal of the following procedure is to detect IFN-γ production at the single-cell level to assess the cell-mediated immune response directed against Varicella-Zoster virus, or VZV. This is accomplished by first coating the wells of a 96-well ELISpot plate with IFN-γ capture monoclonal antibody. In the second step, the cells are plated and stimulated with VZV antigen. Next, the cells are removed, and the captured IFN-γ is labeled with a detection antibody. Ultimately, IFN-γ production and the magnitude and the quality of the cell-mediated immune responses directed against VZV can be quantified.
The main advantage of this technique over existing methods like PMHC tetramers and intracellular cytokine staining is that interferon gamma ELISpot is a robust, reproducible, and informative assay that can be performed on venous blood samples using relatively low-tech equipment such as incubators, centrifuges, and dissection microscopes.
To prepare the plates for the procedure, begin by adding 20 microliters of 35% ethanol to each well of the 96-well multi-screen IP white ELISpot plates to permeabilize the PVDF membranes at the bottom of the wells. After 1 minute, the membranes should become slightly translucent. Immediately manually wash the wells 3 times with 200 microliters of PBS, and then